Hey everybody, we're going to start our notes on 11.2, and that's going to be covering cell division. So when we talked about cell division last week, we focused on why our body needs to actually go through this whole process. And you guys came up with the three reasons of replacement, healing, and then growth. Replacement is just when our cells have died or our cells are being worn out. When we need to heal ourselves, so when we get a cut or a scrape, we have to make new cells to replace those old ones. And then finally, when we grow and develop, like we said last week, you're not the same height, the same size as you were when you were 3. So we have to make new cells so that when our skin grows or our heart grows, our liver grows, more cells are being made for that. So we do need to talk about DNA one more time and why it's so important in dealing with cell division. So, like we've talked about, DNA has all of the instructions that the cell needs. It has the instructions on how the cell is going to function, when it needs to be replaced, what's it going to do, how it's going to do it. So DNA plays a very vital role in all of this. Now, when we talk about cell division, DNA needs to replicate beforehand. Remember I showed you the piece of paper? If I rip it in half, it doesn't have all that information. So like we see in the picture here, DNA has to replicate. It has to make a copy of itself first. So once DNA replication occurs, that means that new cell will have an exact copy of all that genetic information. Now, when we talk about eukaryotic DNA, so we have eukaryotic cells. So animals, plants, insects, we all have eukaryotic cells. And when we talk about DNA, it can actually be found in multiple forms. Now, normally we start off with chromatin. Now, when we talk about chromatin, this is when a cell is not dividing. So this is when a cell is just kind of hanging out and acting like, we'll say, a normal skin cell. Now, if we look at the picture down here, chromatin is that uncoiled DNA structure. So it's kind of in that relaxed, loosey-goosey form. I think of chromatin and I think of the N in chromatin and think noodles, okay? Now, when a cell starts to get ready to divide, it's very hard if our DNA was in this chromatin form. Think about if I had just like a bunch of string in my hand. It would be hard for me to copy it. So what our cell does is that it starts to take the form of a chromosome. So what's happening in that stage, we get this little orange guy here. A chromosome is when DNA starts to wrap itself up and starts to condense itself down. And then when we do this, it makes it a lot easier to copy. So if we look here, it's a lot easier to copy this orange chromosome than it is to get all of these like stringy noodles or stringy DNA and to copy all of that information. So when DNA condenses down into this chromosome form, it can then make a copy of itself. And when we have a copy of itself, we form what's called a replicated chromosome. So that means an exact copy. So we have one side of this X and then the other side of the X. So those are two chromosomes, and if you can see, they are identical. Here's just another picture. We have our chromosome. It's duplicated, okay? Now, holding it together, you see this little white dot here? That's called the centromere. And that is like a little band in the middle that kind of holds it together because, again, we want them to stay together before we divide because we don't want them to be separated. Now, because it's science and we like to call things multiple names, each one of these sides is actually referred to as a sister chromatid. So I like to think of them as sisters because they're identical. They have the exact same information, okay? So these would be like our sister chromatids. We eventually will separate them into those two new cells. So how do we go through this whole part of cell division? Well, it's called the cell cycle. And with the cell cycle, it's a bunch of different things happening. So our body cells, like our brain cells, our skin cells, our heart cells, our liver cells, every single cell in your body, with the exception of sperm cells and egg cells. Those go through a different process. But every other cell in your body goes through the cell cycle. And what it does, it grows and then it divides into two identical cells. As you can see, as the little GIF over here, they divided. We got this cute little guy here. He starts dividing in half and he will make two identical cells. Now, with the cell cycle, there's two main stages. The first phase is the non-dividing stage, which is known as interphase. And there's actually three sub-phases to interphase: G1, S, and G2. Now, we've already mentioned S phase before, and that was when DNA replication was going to be occurring. So, interphase is going to be the longest thing because in this phase, we have a lot of stuff happening. Our cell is growing, our DNA is replicating, and we're copying a bunch of organelles like mitochondria and ribosomes and all that fun stuff. The second stage of the cell cycle is the dividing stage, which can be known as the M phase. This is mitosis and cytokinesis. Now, in mitosis, there's prophase, metaphase, anaphase, and telophase. The last part is cytokinesis, and that is this part right here, where it's actually splitting in half. Now, in order to remember all of this, we have a little saying, because of course we do. I passed my algebra test. I is for interphase, P is for prophase, M is for metaphase, A is for anaphase, and T is for telophase. I passed my algebra test. I recommend you write that down. All right, now let's talk about interphase. So if we look here, we can see interphase is this big green section here, okay, this big green arrow on the outside. We have G1, S, and G2. Then we have mitosis down here and cytokinesis, a very small phase because this is happening very quickly. But again, this top one here, this G1S, G2, this interphase is going to be the longest. And it's actually 90% of the cell cycle because so much stuff is happening here. Now, when we talk about G1 phase, this is where the cell is going to do most of their growing. So we know that cells need to stay small in order to be the most efficient. We did a lab on that. We saw that. However, in order for us to have room for our replicated DNA, our copy of DNA, and for all the extra organelles that we're making, we need a little bit more room. So the cell does have to grow slightly. In S phase, again, that's our DNA replication phase, this takes a while because, again, like we said when we talked about DNA last chapter, we want to make sure that our genetic information has all the right things, all the right steps, all the right nitrogen bases, and that's where that DNA polymerase comes in. The final is G2, and that is just preparing for M phase, meaning all the organelles are present. We have duplicates of everything. So I think of it as getting ready 1, DNA, and getting ready 2. All right? After all of these phases in interphase, then we can move on to M phase, which is the stages of mitosis. All right? Now, in M phase, we have two main stages, mitosis, and mitosis means just it is the division of the nucleus. And there are 4 phases in mitosis. Okay, you kind of see here prophase, metaphase, anaphase, and then the last one is telophase. Now, if you notice, it didn't split yet, and that's the whole point of the cell cycle. But here we see cytokinesis, and that's the second stage of the M phase. Cytokinesis is when that cell actually divides. Cyto means cytoplasm, or it's short for cytoplasm. Okay, and kinesis is that division. So, when we talk about M phase, again, we're not focusing a lot on mitochondria and ribosomes. We are focusing on the division of the nucleus. And this is important because, again, DNA can be found in our nucleus. Okay, the first phase is going to be prophase. And on your paper, probably underneath the picture or off to the side next to prophase, I would like for you guys to write prepare. So the first thing we are going to see here, we have a bunch of stuff moving, disappearing, getting ready. Okay. So the first thing you'll notice is that that circle in the middle is disappearing. That is our nucleus. That is disappearing right there in the center. The second, if we notice, we have that noodle-like DNA, that stringy DNA, but it starts to condense down. And what it's doing, it is condensing down into that chromosome state. It is replicated already, so it's going to start to form those X's like we saw a few slides back. Okay, now it says it consists of 2 sister chromatids. That is just referring to the X, the one side of the X and the other side of the X. Okay, the last thing is that the centrioles are going to start to form spindle fibers. Now on your picture, I want you guys to label the centrioles, and that are these little tic-tac looking like things that are going to the side of the cell. Okay, they kind of look like almost a little T. Please label those on your paper. So this is prophase, and we say prepare because, again, everything is preparing. The nuclear membrane's disappearing, our chromosomes are condensing, and our spindle fibers are starting to get made. The second is going to be metaphase. Again, next to the word metaphase on your paper, please write middle. So we can kind of see why we're saying middle, because what's lining up in the middle are chromosomes. So chromosomes are going to then move to the middle of the cell. Now, it's a little bit easier to see in this picture, but we can see the sister chromatids. So if we look at the X's, again, those are our replicated chromosomes. One side of the X over here is a sister chromatid, and the other side is a sister chromatid. We also are starting to see those spindle fibers reach out. And the spindle fibers are reaching out and they are grabbing on at the centromere. And remember a few slides back, I said it's like that little button or that little belt in the center here, like that little green dot there, the little pink dot here. That's the centromere. So the spindle fibers are reaching out to grab onto those because we need to pull them apart, which leads us to our third phase of mitosis, and that is anaphase. So next to the word anaphase, you are going to write apart. As you guys can see, there are several things happening to our cell. First, that cell is getting longer because, again, we are going to be cutting that sucker right in half in just a few stages. So the cell needs to get a little bit bigger. Our sisters, they're being pulled apart from each other. They're going, no, sister, come back. Don't leave me. Look at them reaching for each other. Ah! So the sister chromatids are being pulled apart. So those spindle fibers, it's almost like a little fishing pole. It's like reeling it in. And then if you notice, we now have the same number of chromosomes on each side. I have a green, a pink, a purple, and a blue. On this side, I have 4. And I have a green, a pink, a purple, and a blue on this side. The last phase is going to be telophase. And I like to say, ta-da, because we are almost done. Okay, it's like, ta-da, look at what we've made. Now, there's a lot of stuff happening here. We see it's being pulled a little bit farther apart. Those spindle fibers are starting to break apart. We don't need them anymore because they already did their job of pulling them apart in anaphase. The nucleus, that nuclear membrane, look at that, it's coming back right here, keeping that DNA safe because, again, our genetic information is going to give our cells all those instructions and we want to keep them nice and safe. So the nucleus is going to reappear. We have one more thing happening, and that's right in the center there. Let's take a look at this picture. Watch what happens to our cell here at the top and the bottom. It starts to pinch. So, and I almost forgot. Our chromosomes are going to uncoil back into that chromatin, and we can see that happening here. As the nucleus starts to reform, our chromosomes are going, okay, we can relax. Now we can go back into that relaxed state of our noodle stringy form. So again, those are our 3 main things. The last thing that I want you guys to pay attention to is that it's starting to pinch at the top and the bottom at the very end of telophase, because that leads us into our last set, and that is cytokinesis. So if you notice here, our animal cells and plant cells. So in an animal cell, like we saw just a minute ago, it was pinching in telophase. An animal cell, it pinches it in the middle until pop, you got two separate cells. They both have a nucleus in the center with our DNA in that chromatin noodle state relaxed form, okay? A plant cell, though, has a cell wall, so it can't pinch in half. However, it can form what's called a cell plate. What that means, it's forming another cell wall. Okay, like if we wanted to split this room in half, we can't pinch it in half, but we can put another wall up. So, overall, the whole idea behind the cell cycle, that's this bottom part here. Two separate identical daughter cells are formed. We started off with one, we ended up with two. And we say that they are identical because, again, each of those cells has the same number of chromosomes as the original cell, which we tend to call the parent cell. So if my cell originally had 5 chromosomes, that means that when I created 2 new cells, each of them will have 5. Here's just a quick little video of a cell going through the cell cycle. Okay, we can see they're lining up in the middle, they're being pulled apart, and then 2 cells were made at the end. All right, if you have any questions, please make sure you are rewinding and getting all this information written down. If you have any additional questions, please make sure you ask.